Here we see a nerve cell, also called a neuron, and obviously it's going to be definitely going to be one of our most active cells. And on the left-hand side, we can see the dendrites going through the soma, or cell body. We can see the axon running from the cell, and then we can see the terminal ends of the axon, where it would be releasing the neurotransmitters. Now it's going to need to make those neurotransmitters, so we see our rough endoplasmic reticulum as part of the way in which it actually makes the neurotransmitters. We would also see a lot of mitochondria in this because this is a very active cell using quite a bit of energy. And then our bottom picture is one of our gametes, the sperm, and the sperm has a lot of mitochondria because it actually has to be energetic, propelling the flagellum, and but mainly its main job is to bring the genetic information from the male to the egg so that we can then fertilize the egg and form a zygote, which has the full number of chromosomes. Now, the sperm and the egg both have what we call a haploid number, meaning that whereas every other cell that has a nucleus in its DNA have the full number of genetic information, the full number of chromosomes, which we call the diploid number, the sperm and the egg only have half the number, so when they are fertilized, they restore the full number of chromosomes.
One of the major physiological principles that we want to make sure we're clear on, because it's so important, is membrane transport mechanisms. In other words, how do things move into and out of the cell across that membrane? And basically, we break it down into two types. What we call passive transport, which means there's no energy being utilized. It's usually operating on the principle of diffusion. Or active transport, where there is energy, usually in the form of ATP, being utilized. Since the body is mostly water, and there's so much chemistry going on within the body, especially at the cellular level, then we need to make sure we understand a few terms that will help us understand what's actually happening. And the cell basically is a solution which has two or more components, and usually a lot more. Water is usually the solvent because it's dissolving what's in it, or possibly not dissolving it, but if it is dissolving it, it is the solvent. And then the solutes would be the smaller things, usually electrolytes, ions, those type things, even though it can be even things like proteins, things that are actually in the solution. So that solution is made up of the solvent, usually water, and solutes, the things that are in that solution. And of course, we also need to make sure we still understand where is water in the body. We know that about 62% of the water is intracellular fluid, and of course that's the fluid inside of the cell, which is basically broken down into the cytosol and the nucleoplasm. Even though it's not pure water, it is a solution. And then of course there's water outside of the cell, and we call that extracellular fluid, and we can even term the majority of that as interstitial fluid, or the fluid right outside of the cell. We say that the membrane is selectively permeable, meaning that it will allow some things to pass through but not allow other things. So it's not completely permeable, it doesn't allow everything to go in, and it's not completely impermeable, keeping everything inside or outside. But not everything can travel through the cell membrane in exactly the same way. So we want to look at some of the different ways that things actually pass through this selectively permeable membrane. The driving force of most passive transport, which means there's no energy being used by the cell in the form of ATP or GTP or other things, is the driving force is what's called diffusion. And basically diffusion is the movement of molecules from, or particles from, a high concentration to a lower concentration. Now, basically it does this because these particles are always in motion, so if they're in a high concentration, they're basically bouncing off of each other to a less crowded area. So they eventually then will equalize, even though they'll never stop in their total movement, they will kind of equalize from a high concentration to fairly equally concentrated all over if it's allowed to continue. A couple of other specific types of diffusion in this passive transport process is first osmosis, which basically means water diffusing through the membrane. And water is highly polar, being charged, and it has the ability to pass through pores in the membrane. The other type of diffusion is facilitated diffusion, and facilitated diffusion means instead of particles just passing easily through the phospholipid bilayer, that they actually have to go through proteins that are embedded in that bilayer that act kind of as doorways or act as transport mechanisms that carry the item across the membrane. This slide shows us two of the three types of diffusion that we have discussed. Picture A that says simple diffusion is actually showing molecules going from outside of the cell and the extracellular fluid down a concentration gradient to the cytoplasm that's on the inside. Now, when we say down a concentration gradient, we mean that they are moving from a higher concentration to a lower concentration, which basically is our definition of diffusion. We have certain particles in the body that are fat-soluble or lipid-soluble solutes that have the ability to pass straight through that membrane. Alcohol has this ability. There are certain fat-soluble vitamins. There are cholesterol and cholesterol-based hormones that have the ability to do this. But the majority of things in the body that need to go into or outside of a cell don't have this ability. But there are some things that do. Those that do, this is called simple diffusion. Looking at picture B, facilitated diffusion, we still have a situation where we have molecules that want to move from the higher concentration, in this case outside, to the lower concentration inside. It doesn't require energy, but these particles cannot just pass through the cell membrane. So we have to have some sort of protein that will facilitate their shape, facilitate their size, that will allow them to pass into the cell from the outside. So these proteins are very important, facilitate the shapes, allow things to either move inside or outside. Now the third type that we don't see a picture of, but we will go ahead and describe, is the movement of water across the cell membrane. And of course, this movement of water from a higher concentration to a lesser concentration is called active transport. Is going to be a process that uses ATP, it uses energy, and the reason is because these things are not able to move by diffusion or going from a high concentration to a lower concentration. There's a couple of different reasons. One thing, they actually may need to go, instead of with the concentration gradient, from higher to lower, they actually may need to go from lower to higher. In other words, they need help going in a direction that they wouldn't normally go, and that help is in the form of energy, and that energy is in the form of ATP. Another thing is they can't necessarily go through the phospholipid membrane, so they have to be pumped through some sort of protein pump. We'll be talking about those. That'll be part of one of our forms of active transport, which is solute pumping. The sodium-potassium pump being probably the pump that we'll talk about the most, but there will be other pumps we'll talk about. And then another thing is that sometimes there are things that are way too large even to pass through protein channels. So instead we have to do something called bulk transport, which is usually in the form of endocytosis and exocytosis. Solute pumping, where we're needing to actually pump something usually against its concentration gradient. Again, instead of something moving from a high concentration to a lower concentration, there are times when we need to actually move it against its concentration, from the lower to the higher concentration. And since this doesn't occur naturally, then it does require energy. It's kind of like when you're floating downstream in a canoe, you don't have to paddle, but if you need to go back upstream against the way the water is moving, then you will have to apply energy to get back upstream. So our cells will have to do the same thing. We will have to apply energy when we're going to move things against their concentration gradient. Here we see one of the main active transport processes, which we're seeing our sodium-potassium pump. And notice in this, first thing I want you to notice is that we do have this protein embedded in the phospholipid membrane. This protein is a pump. It is active transport. Therefore, looking on the left-hand side, you'll notice that we are phosphorylating the pump by breaking off a phosphate from ATP, turning it into ADP. And when we do that, what we're going to do is we're going to exchange 3 sodium ions for 2 potassium ions. We're going to pump them not with their concentration gradient, but against their concentration gradient. Basically, we're going to keep the concentration of these ions from equalizing on both sides. So we have to utilize energy to pump more out, even though more already exists outside, at least more sodium, and more potassium back in, even though there's already more potassium inside. We know it's naturally, by diffusion, going to want to move from the higher concentration to the lesser. So to keep that from happening, we have to use these sodium-potassium pumps to keep the unequal concentration so the movement continues. But remember, the key is this does use ATP energy for this transportation process to occur. Bulk transport is an act of transport that is, we're removing very large materials either outside of the cell through a process called exocytosis, or into the cell through a process called endocytosis. Again, most of these things are being carried within a membrane, a phospholipid vesicle-bound membrane, and again, we're taking large things. So this is really a tremendous energy user. It's like if I had an elephant standing in a room and it can't go through the doorway, I may have to bust out the hole. Here we see that, talking about bulk transport, exocytosis, releasing materials from the cell, endocytosis, bringing large things in, and we even have different types of endocytosis. Something that's very common that we see in white blood cells, cells that we actually call phagocytes, it's called phagocytosis, basically where the cell surrounds something by its membrane, pulling it in. And we even have a variation of that where the cell is bringing in a fluid, a water-based fluid usually, called pinocytosis. Here we see an example, a picture that actually shows both endocytosis and exocytosis. If you start on the left, notice how the cell membrane actually has the ability to surround some sort of large substance that we're wanting to bring in the cell for whatever reason, and it can be for many different reasons, but we're bringing in something from outside inside, we're surrounding it, forming a vesicle out of it. That process is called endocytosis. A lot of times when we bring something in, we actually have to open up that phospholipid envelope, so to speak. So sometimes we'll actually fuse a lysosome to that vesicle to open it up, to either release or to alter the contents, or even to trigger a change and then a release of the substance, as we see going on in this picture. And so on the right side of the picture, we actually see near where it says number 3 that we're actually transporting something from the cell to the outside. So again, that would be an exocytosis. So again, endocytosis bringing substances in, exocytosis taking substances out. They're both active transport because they both require a tremendous amount of energy for this to occur.
Okay, every cell has a life cycle, and that life cycle we can kind of divide up into the cell's normal life being what the cell is, carrying on its normal metabolic processes, and that's called interphase. But then if a cell is triggered to where it needs to divide, needs to become 2 cells from 1 cell, then the cell will have a growth period, and it will have stages that actually prepare it for division. We talked about those in lab, but we may talk a little bit more about what actually goes on in that time before the cell actually divides. Now, when a cell divides, it actually kind of does this through both mitosis, what's going on with the actual DNA and chromosomes, and cytokinesis, which is where the cell actually splits in 2. And of course, the cells do this, they do divide, so we can produce more cells, or so they can have a repair process. Now, not every cell divides after it reaches maturity. Every cell has the potential to divide at least sometime in its life cycle, but there are some cells that continually divide all the time. We continually need to replace them. There are other cells that once they're mature, there is no more division in them. And so we tend to see them lose their centrioles if they don't need to divide anymore. Whereas some cells, such as certain skin cells,